For any assignment-related queries, call us at +1 678 648 4277. You can mail us at info@nursingassignmenthelp.com or reach us at https://www.nursingassignmenthelp.com. Question 1. Gluconeogenesis number 1, Luis. Gluconeogenesis converts various products into glucose for the body to use. A. Write the balanced equation for gluconeogenesis. 2-pyruvate plus 4-ATP plus 2-GTP plus 2-NADH plus 6H2O-glucose plus 4-ADP plus 2-GDP plus 6-pi plus 2-NAD plus 2H plus. B. Why is the equation for gluconeogenesis not the reverse of glycolysis? Under cellular conditions, 3 reactions of glycolysis are essentially irreversible and will not happen in the reverse direction. C. Provide the workarounds that gluconeogenesis takes to overcome the rate-limiting steps of glycolysis. OAA PEP via PEP F1,6BP F6P via F1,6B POSA G6P glucose via G6 POSA. Nursingassignmenthelp.com. D. What is the net result of nucleotide triphosphate production/hydrolysis if gluconeogenesis and glycolysis occur at the same time? How do cells favor gluconeogenesis or glycolysis such that they are mutually exclusive if there are many steps that overlap between the two processes? If gluconeogenesis and glycolysis take place at the same time in a cell, the net result is the hydrolysis of 2 ATP and 2 GTP molecules. The way cells ensure that these two processes are mutually exclusive is by inhibiting the enzymes involved in the rate-limiting steps for each process. E. Avidin, a 70-keta protein found in egg whites, binds biotin with a very high affinity. What step of gluconeogenesis would be inhibited if avidin was introduced into the system? Pyruvate carboxylase uses carboxybiotin to carboxylate pyruvate in the first step of gluconeogenesis. The presence of avidin would inhibit this reaction. So technically all of gluconeogenesis is inhibited, but mentioning this reaction is enough. Question 2. Gluconeogenesis number 2, Sandhya. Nursingassignmenthelp.com. Hyperglycemia, or high blood sugar levels, can lead to several physiological complications. As a researcher at a pharmaceutical company, you are searching for new drug targets to treat hyperglycemia. Which enzymes in gluconeogenesis would be good drug targets, and why? PEPCK, FB POSA, G6 POSA * pyruvate carboxylase is important to gluconeogenesis but is not uniquely involved. Inhibiting it will disturb other essential pathways. These enzymes are key to the progress of gluconeogenesis because they catalyze the irreversible steps, which correspond to large changes in free energy. These enzymes are unique to gluconeogenesis and will not disrupt glycolysis or other essential pathways. 2 patients with severe hypoglycemia come into the ER. You suspect there are deficiencies in their gluconeogenesis pathways and do some tests. Cytosolic biotin levels, mitochondrial biotin levels, cytosolic GTP levels, cytosolic ADP, and AMP levels. Patient 1 normal low slightly high slightly low. Patient 2 low normal normal very high. Nursingassignmenthelp.com. B. Which key enzymes in gluconeogenesis are likely affected in patient 1? Explain how the data supports your conclusion. PC. It converts pyruvate to OAA in the mitochondria and requires biotin as a cofactor. Low biotin levels in the mitochondria will prevent PC from catalyzing this reaction. The slightly high GTP levels are likely a result of not enough OAA being generated. A phosphate from GTP is necessary in the conversion of OAA to PEP by PEPCK, so low OAA levels will correspond to higher GTP levels. The Slightly low ADP/AMP levels are likely due to the defective gluconeogenic pathway, which normally yields for ADP molecules. C. Which key enzymes in gluconeogenesis are likely affected in patient 2? Explain how the data supports your conclusion. The very high cytosolic ADP and AMP levels serve as feedback inhibitors to PC, PEPCK, and FBPASA, which are key enzymes in gluconeogenesis. D. Describe a feedback mechanism within the gluconeogenesis pathway. NursingAssignmentHelp.com. High PEP levels activate FBPASA. This is called feed-forward activation, as PEP is a product early on in gluconeogenesis. AMP and ADP inhibit PC, PEPCK, FBPASA as negative feedback. E. Why is compartmentalization necessary in gluconeogenesis? In general, compartmentalization maintains different ratios of metabolites, like ATP/ADP or NADH/NAD+, to favor certain reactions others. In the case of gluconeogenesis, the mitochondria has a high ATP/ADP ratio which favors the pyruvate carboxylase reaction to convert pyruvate to OAA. The ATP is required to phosphorylate bicarbonate to then interact and react with biotin so that pyruvate can be carboxylate. F. List and provide structures for 3 different products that can be used for gluconeogenesis. NursingAssignmentHelp.com. Question 3. Glycogen synthesis and degradation, Sandhya. A. Draw the pathway for glycogen synthesis as catalyzed by glycogen synthase. Make sure to include any intermediates/cofactors/organic compounds that are necessary to drive this reaction. NursingAssignmentHelp.com. B. How many net ATP are required to add 1 glucose molecule to a glycogen chain? 2 ATP, 1 ATP to regenerate to UTP, and 1 ATP to phosphorylate glucose to get G1P. Glucose, G6P, G1P. NursingAssignmentHelp.com. C. Describe how phosphorylation regulates the active and inactive states of glycogen phosphorylase and glycogen synthase. Phosphorylation of glycogen phosphorylase, active phosphorylation of glycogen synthase, inactive. D. How would each of the following affect an individual's blood glucose levels through glycogen synthesis or degradation? Give a brief explanation. 1. Overactive phosphodiesterase. Lower blood glucose levels, PDE would deplete CAMP levels and prevent pKa activation. If pKa is not activated, it cannot phosphorylate glycogen phosphorylase to activate it. Glycogen phosphorylase releases G1P from glycogen, which can enter the bloodstream. 2. High epinephrine levels. Higher blood glucose levels, epinephrine leads to the activation of pKa, which phosphorylates glycogen synthase to inactivate it, and phosphorylates glycogen phosphorylase to activate it. This dual action promotes the release of glucose from glycogen and prevents other glucose molecules from being incorporated into glycogen. NursingAssignmentHelp.com. 3. Drug that increases insulin receptor affinity for insulin. Lower blood glucose levels, insulin inhibits GSK. GSK normally promotes the inactivation of glycogen synthase by phosphorylating it. Inhibition of GSK thus prevents the inactivation of glycogen synthase and increases glycogen synthesis. E. Glycogen is a polymer of glucose with both reducing and non-reducing ends. Draw an end from a glycogen polymer where glucose monomers are added or removed. You only need to show 2 glucose monomers in your glycogen polymer, but clearly label the end and add a dashed line where to the bond would be to the next monomer in glycogen. State whether the end you draw is the reducing or the non-reducing end of glycogen and explain briefly why that end